Okay. So this work has relied very heavily on Ron. Andrew Jansen replaces Gary Bouchon and will be responsible for all of the scanning electron microscopy, other than the tabletop that Ron uses. And then Ramon, obviously, is my partner in crime with the virus side of things. So here we have the image from Gary of Fructophilus that has been widely used. And on the bottom left, the yellow circle there marks the presence of a mite among the trichomes. And at this magnification, it's, well, at least I can't see the mite, but that's where it is. So the sepals is the preferred location for the mites in the flowers, as has already been stated. So let me move on. So these are the objectives that we are addressing. So first, the need to establish colonies of both virus-free and of RRV viriliferous mites on double knockout roses. So we have some field-grown knockouts that originally came from the University of Maryland campus, which was, that planting was to be removed. So we grabbed some of those plants and transplanted them to the South Farm at Beltsville. And I think that at this point, that was probably 6 years ago, and they are still at least half alive. We have transplanted in between them in succeeding years some various healthy plants out of the greenhouse, none of which are now currently surviving. We obtained more plants this fall from another location and earlier in the year from Washington, D.C. So when we wanted to obtain viriliferous mites from those field-grown plants, we found that there were also significant populations of predatory mites, which made it difficult to find large numbers of mites to transfer. So we put a cage over the plant with the most growth and introduced ladybugs to control the predatory mites, which they have done very nicely. Then we have been able to use flowers from that cage plant either to attach to flowering double knockout roses in the lab and cover those with clear autoclave bags to both contain the mites and maintain humidity. And we have observed systemic symptoms several weeks later. And in fact, the first plant that showed any symptoms, I first noticed it at about 10 weeks after inoculation, but at that point it was on multiple leaves on the plant, so I suspect it was probably visible 6 to 8 weeks. So here we see the cage plant out in the field. So this is a tent that I think is normally sold for family outings to the beach, where you can keep the bugs off and have some shade, but it works very nicely as a cage to keep ladybugs in. So we added several thousand ladybugs to this cage and supplemented it a couple of times. And then on the right, you can see the ladybugs actively crawling all over the plants, and they did a pretty good job of cleaning up the predators. Then here you can see a greenhouse grown double knockout, and attached to it is a bud from one of the field infected plant enclosed in an autoclave bag. And here you can see another one with flower bud and stem attached just below the bud of the plant that we are attacking. And we've maintained that on a light stand in the lab. So that's around 70, 75 degrees, very variable humidity. At least outside the bag, very variable humidity. We have needed sometimes to shake the bags to get drops of water to go down so that we can see what's going on in there. because the humidity is certainly pretty high. These are symptomatic leaves from that double knockout plant, the first double knockout plant. And this was about 10 weeks after the flower attachment. And obviously the symptoms here are quite different from what you typically see in the field. But I talked to Yanis about this at the APS meeting, I guess, and he said this is the sort of thing that they had observed also in greenhouse or growth chamber. So we have also used clip-on cages with a 25 micron mesh to contain the mites. So we use a dissecting microscope, add the mites into the lower half of one of these closed cell foam clip-on cages, between 5 and 20 mites at a time. Or if the mites are difficult to find, but we can see that they are there on the sepals, we have detached sepals of rosebuds and put 2 or 3 sepals in the cage instead. Immediately after putting the mites in the lower half of the cage, we'll lay a piece of white paper over the top just to give them a little shade and to be able to move that cage to where we will attach it to the plant, and then take that to the plant and assemble that with the mites in the lower half on the underside of the leaf, the upper half of the leaf cage placed over the leaf and pinned into place. And then we have supported the cage and the leaf that it's on using a bamboo skewer either side of the leaf and a twist tie to attach the cage to the skewers, as you can see here. So typically we have put either 1 or 2 cages on a plant. These cages come with a mesh size that is intended to keep thrips in place, but is which mites would be able to walk through. But the manufacturer makes this with a layer of waxed paper on top of the cage, and they provide the mesh separately. So we were able to just cut circles of a 25 micron mesh and glue those, attach those just by peeling off that protective coating and adhering the 25 micron mesh to the cage. So these have worked very nicely. They are quite lightweight, and on a more woody plant with sturdier leaves, they might not need any support. But here on the roses, the relatively soft tissue, the leaves would bend down too much. So we use the bamboo stakes to hold them up. So on the left here, you can see one of the, this was not the first plant, but a later plant. So here, while we had a cage still attached to the leaf, we could see obvious symptoms starting to develop systemically. Infected plants showing symptoms have then been transferred into these cages in the greenhouse. We are now making new cages which have a larger area of the 25 micron mesh on each side as ventilation ports that through which the mites cannot escape. Also, to prevent mite escape, these cages are standing in a container with filled with water, so that there is a water barrier at the bottom of the plastic, so that the mites cannot escape that way either. And as you can see on the right, the containers, the plant sources here are sitting on bricks to keep them just above the level of the water. And for these cages, we have introduced a tube from the outside so that we can water into the tray as necessary. That's not necessary very often, because with the water inside the base of the cage and the humidity rate retained inside the plastic, there's relatively little water loss. When we do put water into the sources here, I poked holes at about 1/4 of an inch from the base of the saucer so that we cannot put too much water in. Any excess will drain out, so the plants don't become waterlogged. So now, so we now have colonies of we have initiated colonies of rose rosette free mites by both floral attachment and by clip cage transfer in autoclave bags on plants in the lab. These have not yet been transferred to the greenhouse. And our observations support what everybody else has been saying so far, that uninfected plants have very much lower mite populations. So it's quite a struggle to find enough mites to put in a cage. So we have used floral attachment as well. So the purpose of these rose rosette free mites is to be used for acquisition and transmission tests on the core set of roses, with acquisition from the core set plants, which will have been initially graft transmitted from infected double knockout plants originally infected by mites. The transmission tests from the core set will be by mites raised on rose rosette infected plants of the core set, that is. They'll be fed in cages on graft inoculated plants for 14 days and then transfer the cages with the mites to test plants of double knockout for 30 days following that, prior to miticide treatment and following symptom development and RT-PCR testing. Transmission tests to the core set will use rose rosette virus infected mites from double knockout plants as the source hosts, and a 30-day transmission access to the core set plants. And then these will be tested, both types of these experiments will be tested by RT-qPCR at 60 to 90 days from a minimum of 2 locations on each plant. Here you see a double knockout plant grafted with 2 shoots from a rose rosette infected rose and placed in a clear bag to maintain the humidity. So for these grafts, we have used a steep slant cut on both the stalk and the scion, bound those together with parafilm, and then held them with a grafting clip. And then used a bamboo skewer both to support the scion and to help keep the bags spread open so that we don't get them sticking together at the top and too much humidity without airspace. So we will also be grafting like that to each of the members of the core set so that we have so that we can compare the speed of movement of virus in the different genotypes of the core set, and also so that we can have graft inoculated mites, graft inoculated members of the core set to which we can add rose rosette free mites to do acquisition testing. So in order to assess mite reproduction, body size, and feeding behavior, which is another thing that somebody was asking a question about earlier, we will be using 20 mites contained on detached leaves and separately on flowers and maintained under a 16-hour photo period, and then count the numbers of adults, immatures, and eggs on leaves at 7 and 14 days after attachment. The mites on the flowers will only be counted at 14 days because it's necessary to dissect the flowers. At the end point of the feeding studies, the tissue samples will be examined to look at the numbers of mites, the location and appearance of feeding holes, and to compare the number of feeding holes per mite so that we can get an average count of the number of feeding holes compared to the number of mites, so that we can make a comparison between different genotypes. We have some prior observations that indicate that different rose genotypes may have differential responses. When we initially looked at the mite feeding sites, we found that some feeding holes had a raised appearance right around the feeding hole, which was reminiscent to me of the saliva tube that occurs on many plant hosts when aphids feed. But we don't think that that is a saliva tube from the mites, but it suggests a possible plugging of the feeding holes, possibly by gelled saliva or more likely by a host defense response of callose laydown, which is a common host defense mechanism. So basically, we're interested in looking at the effect of the rose genotype on the mites. And by feeding both healthy mites and rose rosette carrying mites to determine how much effect the virus has on mite reproduction, numbers of feeding holes, and other things like that. So as I just said, the feeding hole numbers will be tallied and compared with the number of mites. So host defenses may cause the mites to move from one feeding hole to another. We have seen many feeding holes in close proximity on the same cell. We suspect that this is due to the activation of some host defenses, or possibly that the mites need to move more frequently because they need more salivary excretions, which are stimulated by fresh feeding sites. So this will be determined by cryoscanning electron microscopy, and that will be done primarily by Andrew. And then we will also have some material embedded for ultra-thin sectioning and for transmission electron microscopy to look at, hopefully to be able to find sections through feeding holes, feeding sites, to determine whether there is host cell wall thickening or other reactions immediately around the feeding site, and whether that differs between different rose genotypes. So hopefully we will be able to address through these various experiments quite a few of the questions that have been raised earlier and that we were discussing at the virology and genetics meeting breakout last night. So this is just a shot that shows you Phylacoptes fructiphilus, different life stages. So the A is the adult, N1 and N2 are the two nymphal instars, L down the bottom here is a larval form. There are multiple eggs scattered around the view. The X marks exuvia or shed skins. These ones from one of the nymphal stages. This may be from the nymph to the adult stage, possibly from the adult that is right here. And this is what I would like to draw your attention to, because here there are many feeding site holes in close proximity to each other. So they don't need to pick up and move a long distance away to feed. A lot of the feeding sites can be very close together. Oh, I missed pointing out one thing here. Here and here and a few others there are spermatophores deposited by the male and picked up by the female. So I don't think Ron mentioned that aspect of biology, but there is no mating as such. When a female arrives on a new plant, when she is not fertilized, she gives rise to male young, and all of the eggs would be male. But once this female has spermatophores deposited by the male, then she picks those up and produces exclusively female mites. So I think that covers that. Two different views of the mite, and thank you for your attention, and I'm open to any questions. So I'll leave the images up in case anybody wants me to go back to anything.
Hey, John.
Yeah.
Mark, I got a couple of questions for you. On those, when you were talking about those feeding holes, and and the feeding holes getting plugged,
yeah.
is the mite moving because host defenses are walling off its feeding hole, or can it come back to an old feeding hole if it's not plugged? Have y'all observed anything about that type of behavior?
We haven't done the sectioning on that yet.
Okay.
That's something we need to do. I suspect that there is a host defense and that the holes are getting clogged by deposition, maybe of callus. And that means that the mite has to up sticks and move a little.
Okay.
But the host, if it is host defenses, it's not broadly effective across the whole cell, because they're able to go and feed, and you can observe mites feeding on cells where there are many previous holes. So if they're actively feeding, they are obviously not inhibited at a fresh site. But
okay.
we need to see thin sections to answer that question. We have seen some evidence of wall thickening with a different mite, with the lychee mite.
My other question has to do with that picture where you had the grafted plant in and not in the plastic bag. In the picture that you showed, all the symptomatic host tissue was above the graft.
Correct.
After you did the graft, did any symptomatic shoots come from the plant, from the rootstock below? And if so, how long did that take?
Hasn't been long enough to be able to see symptoms on those yet, or I have not seen symptoms on those yet. I hope to be able to answer that before too long. I'm trying to think when I've made the first grafts. And I took the picture without a bag because that was the best way. I did that immediately after grafting, and as soon as I'd taken the picture, I put a bag over to maintain the humidity.
Okay. I was just curious about that.
And having raised the question of humidity, with those large cages in the greenhouse, we are probably not getting enough air exchange. The humidity in there is probably higher than desirable. And also the temperature, we are making new cages with a larger area of the 25 micron mesh, so that we can get better air exchange and reduce the temperature. We have put a temperature probe inside one of those cages, and on a good day in the greenhouse, that can reach 90 degrees.
Well, that was going to be my last question. When you put the bag on and that humidity spikes like that, are you seeing any issues from diseases like powdery mildew that would not normally be on knockout roses in the field, but when we have put them in high humidity situations, all of a sudden we start having mildew problems that we didn't have before? Have you encountered that? And if you have, what are you doing about it?
We haven't seen mildew start inside the bags. We have had some issues with mildew in the greenhouse, and if you recall, I called you.
Yeah. I remember that
weeks ago, and that was in the greenhouse,
but not in the bags.
Not in the bags.
Okay.
Now that I did find one plant that I was about to inoculate that had mildew on it, so I set that one aside and blasted it with mill stop and used it later after the signs of mildew were gone.
Thank you.